Okay, so we're going to then continue with our discussion of chapter 4 about the organelles in the eukaryotic system. And then we will move into chapter 6, which is microbial growth. So one of the organelles that these eukaryotic cells have is this rough endoplasmic reticulum. The rough endoplasmic reticulum is made by a series of membranes that are folded, and there is a space between these membranes that allows the transit of messenger RNA, and the messenger RNA is going to be read by these ribosomes. So the ribosomes are attached to this rough endoplasmic reticulum, and then you make a protein out of this. So functions then of rough endoplasmic reticulum is to make proteins, protein synthesis. And then we have another type of endoplasmic reticulum in which you also have membranes that forms like little tubules internally, and this membrane of the smooth endoplasmic reticulum doesn't contain ribosomes, and the function is to synthesize lipids like steroids, and as well to detoxify the cells from drugs, alcohol, for instance. So if you look at this transmission electron micrograph of these two organelles, they look a little bit different. So again, both are made of stacks of membranes, but if you see here on the right side, you see these little dots? This represents the ribosomes attached into this rough endoplasmic reticulum. And then here on the left, this little part, you see stacks of membranes, which are all these little lines, but you don't see the ribosomes. So that's a difference then. Smooth produces lipids, this one produces proteins, and this one also detoxifies the cells. So ribosomes are the protein factory of the cells as a majority. So these ribosomes in eukaryotic cells can be 80S or 70S. And if you remember, S stands for set per units, which is a unit of sedimentation when you are centrifuging a sample. So then a difference between the 80S ribosomes and the 70S ribosomes is actually that 80S are larger and they are denser than the prokaryotic ribosomes, which are 70S. Now, where are these 80S ribosomes in the cell? Well, they can be attached into this rough endoplasmic reticulum, or they can be free-floating in the cytoplasm. So these will be called free ribosomes. No matter if it's free or if it's attached, they have the same function, they produce proteins. And then the 70S, which are a little bit smaller than the 80S, and that they are less denser, they can be found in chloroplasts in plants or in animal cells in mitochondria, and as well in the plants. So then, in one of the chapters, we discussed the possibility that some people believe that there was a transfer of bacteria from a long time ago, billions of years ago, from a prokaryotic organism, let me rephrase that tangle in my tongue. So they were saying that apparently a bacteria was endocytosed by an eukaryotic cell, and that bacteria somehow became a mitochondria. or something like that. So that's why we ended up with 70S ribosomes in our eukaryotic cells, which are unique for prokaryotic cells. That is a theory that is called endosymbiosis, but it's just a theory. So now for the Golgi complex. So the Golgi complex, just as these other organelles that we saw, is made out of stacks of membranes that forms a network. So this space is called cisternae, and then or spaces. And through this cisternae, you receive some of these vesicles coming from the rough ER, and then these vesicles move in this direction. And as they move in this direction, moving from cisternae to or cisterna to cisternae or cisterna, you will have modification of the contents. In this case, it's going to be proteins. And then these proteins will be either released outside of the cells or it will stay within the cells. So this is like a distribution center. And that tag that you add to the proteins will tell what is the destination of these proteins that are made by these Golgi. So the ultimate goal is to make these secretory vesicles, and sometimes these secretory vesicles can have enzymes. So a source of enzymes are lysosomes to destroy the cells when the cells burn out. It is the Golgi. So functions of the Golgi: membrane formation and protein secretion. Mitochondria, it is the powerhouse of the cell. In some cells have like a sausage-like structure. It has a double membrane, the outer and the inner. So this is the outer, this is the inner. The inner infolds to form cristae. So these infoldings are called cristae. And then internal to this inner membrane, we have the mitochondrial matrix, where we have several enzymes and we have proteins that helps to make the ATP. So the Krebs cycle or tricarboxylic acid cycle happens in this organelle. And specifically here between the inner and the outer membrane, there is a space where you have chemiosmosis or transfer of hydrogen ions to help to generate ATP. And something before I move on, characteristic of these mitochondria is not only that it has 70S ribosomes, but also has its own DNA. And mitochondria, at least in humans, I don't know in other species, but in humans, we inherit the mitochondria from our mother, not from our father. So there are some diseases that are called mitochondrial diseases that will be, of course, coming from our mom. Organelles that are part of the plants. So we have chloroplasts. So these chloroplasts are specialized to form these carbohydrates and oxygen through photosynthesis. So in photosynthesis, you combine molecules of CO2 with water, and then you have incoming energy from sunlight, and that will lead to the formation of these carbohydrates and oxygen. Right, so here we have an electron micrograph showing you some of these chloroplasts. And then internally they have like these thylakoids, which are like little discs that connect to each other, and then they provide a specific structure to these chloroplasts. Lysosomes, again, come from the Golgi apparatus and store digestive enzymes. So our cells have they have a they have a chance, right, to get damaged. And with this chance to get damaged, some of these can repair, but some of they cannot repair. And they can have these lysosomes that can help to auto-digest the cell. Or maybe some cells, like macrophages, can endocytose bacteria, and then they fuse these vacuoles with the bacteria with these lysosomes. And then you can have a phagolysosome in which you would have the enzymes released to break down the bacteria into smaller pieces. And then some cells can have vacuoles, and these vacuoles can have food, like in the case of plant cells, they have large vacuoles with food, like starch, and then that provides structural support. Other ones may have enzymes like catalase, peroxidase, superoxide dismutase, that can break down toxic forms of oxygen, including hydrogen peroxide. And within peroxisomes, we can also oxidize fatty acids so that we can break down the bonds and generate energy. And then our eukaryotic cells, when they are about to divide, they have a pair of centrioles that, at least human cells, that separates to the poles of the cells, and they organize the mitotic spindle. The mitotic spindle is basically a series of tubules made of tubulin protein that holds the chromosomes in the middle and pull them apart so that each daughter cell will receive equal number of chromosomes. So this is a summary showing you the main differences between prokaryotic and eukaryotic cells. So in terms of size, these ones are minute. So this is the column for prokaryotic cells. They measure between 0.2 to 2 micrometers in diameter. For the eukaryotic cells, typically it's between 10 to 100 micrometers in diameter. So even the smaller eukaryotic cell is 5 times larger than the prokaryotic cells. So 5 times 2, that's 10, right? And then these ones, they don't have a nuclear membrane or nucleoli, while these ones, they do. Prokaryotic cells, they don't have membrane-bound organelles. They do have ribosomes, but the ribosomes are not membrane-bound. These eukaryotic cells, they do have all of these organelles, including Golgi, the endoplasmic reticulum. Flagella, it has 2 protein building blocks. This one can have like the 9+2 arrangement. So it's a little bit more complex, the structure of the flagella in eukaryotes. And then both of them can have glycocalyx. But the eukaryotic cells, most of the time, like for instance, our animal cells, they don't have a cell wall, although of course plants have a cell wall. Cell wall is usually present in prokaryotic cells. It can contain peptidoglycan or pseudomurin in the case of archaea, while in plant cells or fungi, they can have cellulose and chitin. The plasma membrane in prokaryotes, they don't have carbohydrates, and generally it lacks esters like sterols. And in the case of eukaryotic cells, they have sterols, they have carbohydrates, and they act as receptors. Cytoplasm doesn't have a cytoskeleton in prokaryotic cells or cytoplasmic streaming. This happens in the eukaryotic cells, so through this streaming you can distribute molecules, and then you have cytoskeleton that provides the support. The prokaryotic cells have abundant ribosomes, it's 70S, and then in eukaryotes you can have both 70S and 80S. In terms of DNA, prokaryotes have a single circular DNA. For the most part, there are a few exceptions. We will see later on which ones they are. And they don't have histones that wind up the DNA as it happens in these eukaryotic cells. And then you have multiple chromosomes in eukaryotic cells. So human cells, how many chromosomes do we have? 46. And then division of the cells, binary fission in prokaryotic cells, and mitosis in eukaryotic cells. And then for sexual recombination is through transfer of DNA through the pilus in prokaryotic cells, or in the case of eukaryotic cells is through the process of meiosis. And this is a micrograph showing you a light microscopy view of a eukaryotic cell infected with prokaryotic cells. So see how small is the prokaryotic cell, and then this one has a large nucleus, these two, so these are eukaryotic cells. So highlights of this chapter is that bacteria have unique characteristics that are targets for antibiotics, including the presence of the 70S ribosomes, and then as well the presence of a cell wall. And then they can be also subject to immunity and phage infection. Phage is a type of virus that can infect these prokaryotic cells. And then bacteria and archaea have specialized structures like flagella, endospores, and pili. We don't have endospores in eukaryotic cells, of course. And this helps to help the bacteria to survive even after the bacteria has died, because in a way the endospores is a dormant form of bacteria, or it is, not in a way, it is. So another highlight is that while microscopic eukaryotes like fungi, protozoa, and algae carry some processes or have some structures similar to bacteria, many of these cellular processes are fundamentally different. Okay, so that's all for chapter 4. Now let's move into chapter 6. So chapter 6 is titled Microbial Growth. So in this chapter, we're going to see the microbial classification according to the temperature in which the bacteria grows, according to the pH that the bacteria grows, and as well according to the osmotic pressure. So microbial growth in the lab happens in media, and the bacteria form groups, visible groups, that we know as colonies. So in here, you can see a cartoon of a pitcher plate with agar surface, and all of these little circles, white circles on top, represents colonies. Colonies come from individual bacterial cells. So as the bacteria is dividing, it will form a colony. What does the bacteria needs to grow? No, it has physical and chemical requirements. Physical requirements requires to be in a specific temperature that is preferred by the bacteria, and that allows the bacteria to perform its metabolic reactions. The bacteria also requires a particular concentration of hydrogen ions within the environment, so that's pH. And the bacteria has to be in equilibrium with the concentration of ions or solutes in the environment. This is osmotic pressure. So bacteria has developed the ability to survive in harsh environments, and that it is transduced as having, for instance, a cell wall that helps them to resist this, this osmotic pressure. Now, for chemical requirements, well, bacteria requires a source of carbon, so that carbon it will use it to form DNA, to form proteins, for instance, to generate energy. Also requires nitrogen, sulfur, and phosphorus. And requires trace elements. Trace elements are needed only in small amounts. And are necessary for most bacteria. Oxygen is needed for most bacteria, and they may need organic growth factors to grow. So it's basically all of these chemical requirements are a source of nutrients. And the physical requirements help for these nutrients to be assembled into particular structures like proteins, nucleic acids, lipids to form these membranes, and the components, of course, of the cell wall. Let's talk about the physical requirements. So we have temperature. So independently of the type of temperature or the temperature that is preferred by bacteria, bacteria has an optimal growth in the optimal temperature. But it can have a minimum growth in the lowest temperature that it can tolerate, and it can have a maximum growth temperature within a range. So it has maximum growth temperature, so that will be the maximum temperature that the bacteria can grow in, while the minimum growth temperature will be the minimum temperature in which the bacteria can grow in. And the optimum growth temperature is the best temperature for the bacteria. So here is the bacterial classification according to the bacterial preferred temperature. So we have some bacteria called secrophiles. These secrophiles can grow at a temperature that is very low, minus 10 degrees Celsius, and maximum growth temperature of 20 degrees Celsius. So the optimum growth temperature for these secrophiles is 50, somewhere in the middle. And then psychotrophs, they grow between 0 and 30 degrees Celsius with an optimum growth temperature of 20 degrees Celsius. Right? Mesophiles grow between 10 and 50, and they have an optimum growth temperature of 37 degrees Celsius. So these mesophiles actually is the type of bacteria that we have normally in our body, because our body temperature typically is 37 degrees Celsius. So most of the normal flora, the bacteria we have, beneficial bacteria we have in our body, is a mesophile. And same for pathogens. Bacteria can produce infections, they grow at 37 degrees Celsius, so they are pathogens, mesophiles. Can be pathogens, sorry. And then we have, after the mesophiles, bacteria like to grow in higher temperatures, like, for instance, thermophiles. Thermo stands for temperature, and then file for loving. So in this case, they can grow between 40 and 70 with an optimum growth temperature of 60 degrees Celsius. So basically 23 degrees higher than the normal body temperature for humans. So because of that, because they cannot grow at 37 degrees Celsius, as you can see, like the minimum is 40, thermophiles do not infect humans. Hyperthermophiles grow between 65 and 110. And that will be temperature, the optimum of 95 degrees Celsius. 95 degrees Celsius. Boiling temperature of water is 100 degrees Celsius. So these hyperthermophiles are extreme bacteria that can live in the bottom of the ocean, in gushers of water. So here is a graph showing you the different range temperatures for the different groups that we consider. So in this axis is showing you the rate of growth. So whenever you see these peaks in this graph, in these curves, that means that that's the maximum growth. And then, of course, towards this bottom part is less growth. But it's increasingly the growth better as they reach the particular temperature that they can grow in. And this is the temperature in Celsius. So here we have the mesophiles with an optimum temperature of 37 degrees Celsius, but they can grow between 10 and 50. And then hyperthermophiles. They can grow very good at very high temperature, almost at the temperature of boiling water. And here we have a photograph of a biofilm in deep sea in a hydrothermal vent. So here in the deep sea, this hydrothermal vent releases water at high pressure and high temperature, and these bacteria can grow around this. So one of the reasons why we care at what temperature the bacteria grows is because, of course, we all have to eat to live. And then we have to make sure that whatever we eat is free of pathogens. And one way is by cooking properly the food, first avoiding contaminating the food as you're preparing it. But when you're actually cooking the food itself, you have to make sure that you use the right temperature to kill the potential pathogens. And then once you finish cooking and you're going to store leftovers of food, you also have to make sure that you handle well the temperature of the food. That way you prevent diseases. So here we have in this illustration 2 thermometers. This one is in Fahrenheit, and this one is in Celsius. The one on the right is in Celsius. So we have equivalent temperatures between Fahrenheit and Celsius. So we have defined in temperature for handling food what is the danger zone. So the danger zone for handling food in which most microorganisms grows really rapidly will be between 60 to 130 degrees Fahrenheit, or between 15 and 53 Celsius. So if you are preparing a meal, you want to cook it a little bit above even like this temperature away from the danger zone between, let's say, 130 and 150 degrees Fahrenheit, but still has the potential to have slow bacterial growth. So whenever you're prepping a meal, better go higher than this. And then, so let's say that you prepare your meal and then you went above this temperature, which in this case for Celsius will be like around 64, 65. So yes, okay, you didn't have any pathogens there, or you killed any microorganisms. But what when you are going to store the food? Well, the food can go back as it's cooling off into this dangerous zone. And let's say that your container is open and then there is dust there, or someone tried the food and put back the spoon into the food, which is never a good idea, of course. And then some bacteria contaminates the food. So then as it's cooling off, goes back into this danger zone, and depending on how much food you have to cool off, if it's a lot, will stay a longer time in this danger zone. And that will lead to rapid growth of the bacteria. You will see how later on some bacteria can form millions within minutes as they grow. So then you start eating the leftovers and thinking that they are safe, and then you become sick. So that's a problem. Now, at this temperature, some bacteria, like the secrophiles, can survive. Like we have bacteria that can survive refrigerator temperatures. And this is what we call spoilage bacteria. Like they can survive between 30 to 60 degrees Celsius, or between 0 and 15. This is Fahrenheit, and between 0 to 15 Celsius. And then freezing temperature, like below 0 degrees Celsius, well, there's no significant growth of bacteria. But the problem is with these ones, these spoilage bacteria that can spoil your food, and of course you throw it away, that's no problem. But there are some bacteria that are pathogens, like for instance Listeria monocytogenes, that can produce listeriosis. This listeriosis is dangerous for immunosuppressed individuals and as well for pregnant women. Listeria frequently has been found to contaminate dairy products, processed foods like delicatessen, like ham, hot dogs, and all that, and even some vegetables like salads. And that can cause a stillbirth. So that's Listeria monocytogenes. It can survive fridge temperature. So you can purchase a food thinking that it's safe, and no, it's not. So here's an example of what happens when someone is cooling off food. In this case will be rice. Rice can be contaminated with a bacteria, it's called Bacillus cereus. And then if you cook the food, like if you cook rice and you put them into the two different containers to cool off, like if you have here a shallow container that is only 2 inches in depth, while this one it's deeper, it's much bigger, like this one has 6 inches in depth. So of course this one will take longer to cool off than this. And here it shows you the temperature in this graph between minus 10, to 80 degrees Celsius. And here is the time in hours that it takes for this rice to cool off. So, and sometimes can be even when you put the food into the refrigerator to cool. So if you have a shallow container versus a deep container, of course this shallow container will take less longer to cool than this. And because it takes less longer to cool, it will stay within this danger zone, which is between 15 to 45 degrees Celsius. So it would take approximately like, let's say you started with a temperature in the rice of 70, and then you reach 15 within an hour and a half with this shallow container. Now, if you have a deep container, you start at 80 degrees Celsius, and then by the time of 8 hours, you haven't reached this temperature in the rice because it's still like above 15 in the danger zone. So it spends 1, 2, 3, 4, 5 hours in the danger zone, or maybe even more. So what will be, and this is the danger zone, 15 to 43 degrees Celsius. What will be the remedy to prevent this when you have this deep container with a bunch of rice? To cool it off quickly. Or would you leave it there, like if you work in a restaurant, and then serve it to the customers? What would you do to cool it off quickly? No one has worked in a restaurant here. Will the restaurant has only like a big container? That will be the only container that I will have for prepping rice? So then, if they have many containers, what would you do? Put it in a lot of shallow containers, right? And then put it in the fridge to cool off, to take it out of this temperature. And actually, restaurants, guys, they have big paddles like around this size. And of course, they are like this big, the handle, and then the paddle itself is very wide, and inside has a gel that freezes, and then like they have like these big pots, they introduce it and they start mixing it like this to cool it off quickly. And they have the walk-in freezers where they can put the food and rapidly take it out of the dangerous zone. Okay? So, pH. Most bacteria are neutrophiles, so they like to grow between 6.5 and 7.5 pH. And some are acidophiles, like molds and yeast. They grow between 5 and 6. So if you remember the pH of human plasma, blood plasma pH, is between 7.35 to 7.45. So then these neutrophils can grow in our blood. They can infect our blood. And then in the stomach, we have a pH of around 3. So most bacteria, they don't grow in our stomach because the pH it's so acidic, including yeast and molds. They don't grow in our stomach. But later on, we will see in the semester how bacteria can survive this acidity in our stomach and can infect our stomach, and it can even create cancer in our stomach, can produce cancer. Osmotic pressure. So, if you put bacteria in a hypertonic environment, most bacteria will undergo what's called plasmolysis. You saw that in the previous chapter, in which this excess of solutes in the environment where the bacteria is growing will make, through osmosis, pull water of the bacteria from the cell into the external environment, and then the plasma membrane detaches from the cell wall, and that leads to a dead signal in the bacteria, and the bacteria will die. So hypertonic environments like high salt content or sugar causes plasmolysis. In ancient Roman times, they used to pay soldiers with salt, their work with salt. The reason for this is because salt was used to preserve food. There were no refrigerators. And hence, the word salary comes from the word salt. In Latin, salt is sal, or in Spanish. So in many places, they preserve food with using high contents of salt or sugar. And we will see as well later on how some bacteria, they like to live in these conditions. Well, actually it is now. So we have some bacteria that are called extreme or obligate halophiles because they require high salt content or high osmotic pressure. So an obligate in microbiology means that it must have facultative means preferred but can live without. Let me give you a real life analogy. Well, there's people who, once they grow, they reach the age of marriage or having a mate, they must have a mate. And there's some that can stay single, but they prefer to get married. So that's a facultative versus an obligate, right? So we have obligate halophiles. They require high contents of salt, and we have facultative. So that means that they can tolerate high osmotic pressure, but they don't need it for growing. So any questions about what we have seen? I have seen people who put a lot of salt in their food even without trying it before. That's a halophile. Okay, so here is the concept of plasmolysis or plasmolysis. So an isotonic solution of saline solution, it is having 0.85% of sodium chloride, and of course the rest will be water, like 99.15% will be water. And then in this case, water can move in and out of the cells, but the cell doesn't gain any volume. And then when you add sodium chloride solution that is 10%, so it's hypertonic, in this case, you have a lot of solutes outside. This will be the sodium chloride. And then water will move from an area of low solute concentration, which will be the cytoplasm of the bacteria, to the external environment where you have this sodium chloride. And then you will shrink the cell. So the plasma membrane, which is this one in yellow color, detaches from the cell wall. That leads to cell death. The cell membrane has to be near to to the cell wall in order for the bacteria to survive. So this is called plasmolysis. So now let's go and look at how carbon, nitrogen, sulfur, and phosphorus are needed in larger amounts for microbial growth. And we're going to start classifying the microorganisms according to the requirement of oxygen. And we're going to see how some microorganisms avoid injury by toxic forms of oxygen, and how we form, bacteria form biofilms. So, just as humans, we have requirements of nutrients in large amounts, certain nutrients. Same for bacteria or for microorganisms. So one of these main elements that the bacteria needs to form the structures and then to produce energy will be carbon. Carbon is a component of nucleic acids, it's a component of carbohydrates, it's a component of lipids, of proteins. So thus it makes sense that carbon is needed the most. Now, for the microorganisms that they get their carbon out of organic carbon sources, like for instance proteins, lipids, carbohydrates, these will be called chemoheterotrophs. So they get the carbon from the same sources that they use it for. When an animal decays, a human body decays, and is not preserved, bacteria comes and get the carbon out of our amino acids, our proteins, get the carbon out of our nucleic acids, out of our lipids, out of our carbohydrates that we are decomposing, and they use it for forming the same things. So it's like kind of a recycling process. So then these will be the chemoheterotrophs. Now, CO2 is carbon dioxide, right? That's how we abbreviate it, carbon dioxide, CO2. Does humans use CO2? Or we generate CO2 as a waste? The first or the second? Do we use CO2 or we generate CO2 as a waste? It's one or two. Put it with your fingers. Two. So for us, what is a waste? For bacteria, it's a food. They can get energy out of the CO2. And this microorganisms will be autotrophs. So that's kind of their means of energy. So they are kind of independent. That's why they're called autotrophs. So that's a source of carbon or sources of carbon. Now, nitrogen. Where does the bacteria get the nitrogen? Well, from amino acids and proteins. If you remember, for amino acids, we have nitrogen in them. So we have a central carbon, we have an R group, we have a hydrogen, we have a carboxyl group, and then we have an amino group. Every amino acid of the 20 that we have, no matter which one it is, has all this core. The only thing that varies is this, depending on the amino acids. So then, where is the nitrogen? Here. So bacteria can get the nitrogen out of the amino acids that we have in the proteins. So most bacteria can decompose proteins. Another source where bacteria can get nitrogen can be ammonium or nitrate. And elemental nitrogen. N2 is elemental nitrogen. Elemental nitrogen exists in nature, and it's a gas. Bacteria can fix it. They have the enzyme that helps them to utilize this nitrogen. And only few bacteria can do so. Some bacteria can use ammonium or nitrites and nitrates, which will be NO4. So that's basically for nitrogen. Now, bacteria can also require sulfur. Sulfur is an element. It is found in amino acids. Certain amino acids, like cysteine, has sulfur. Thiamine, it's a vitamin. And biotin, another vitamin. So some bacteria can use sulfate or hydrogen sulfide to get their sulfur. Hydrogen sulfide, it's a gas, it's toxic. Sometimes it's formed as part of the emissions, and then they produce like what is called the acid rain. And then bodies, of course, as they decompose, they have cysteine, they they release the sulfur into the environment and bacteria come and utilize it. And there are some certain areas in the world where you have a lot of these, so these bacteria thankfully live there to utilize whatever is toxic for us. So that's another chemical requirement. Now for phosphorus. So phosphorus is part of these elements that can be, can be found in DNA, RNA, and ATP. So and also phosphate is part of these chemicals where bacteria can get their sulfate. So where DNA and RNA are located? In the cytoplasm or in the nucleus? Or both? Well, first DNA. DNA, is it found in the nucleus, number one, or in the cytoplasm, number two? DNA is found only in the nucleus, is number one. And then RNA, can you find RNA in the cytoplasm as well as in the nucleus? Yes or no? Messenger RNA goes from the nucleus into the cytoplasm, so it can be found both, right? That's why it's messenger. And now for trace elements. So, trace elements are not requiring huge amounts, just like as our human diet, and they are inorganic elements. So for instance, we have magnesium, zinc, copper, etc. And this will be a source well, these will be cofactors. So cofactors, enzyme cofactors are required for functioning of the enzymes. So I'm going to make an analogy that helps you to understand what is a cofactor. I know that many of us, we like volunteering work, and we have done a lot. And I haven't seen many people living only out of volunteer work. We all have to do kind of a source of work that pays us. And let's say that you're very altruistic for the company or for the person that you work, and you work for this person for a month, and then the person or the company haven't paid you. And they kind of keep you with the promise that they will pay you. But eventually, 3 months has gone by, and they haven't paid you. Would you work for that company or that person? And let's say it's not your parent or it's not family business. Typically not. I mean, let's say that you don't have some source of help for paying rent and the bills. I mean, you won't work, right? So in that case, what will be the cofactor for you to continue to work? And tell me, what is that unit of currency that will be your cofactor? So what will be your magnesium, your zinc, or your copper to keep working for that company? Don't say only money. What is the cofactor name? What is the money? It's not pennies. They don't exist anymore. Well, they're about to disappear. What is the name of the cofactor that they will pay you to keep working? It's made out of 100 pennies. Still no? It's not 1 peso. Huh? Dollars. Wake up. It's almost time to leave. That's the cofactor for us to work. So enzymes work with magnesium, copper, zinc, depending on what enzyme it is, what it needs. Sometimes calcium also can be a cofactor. So then we have organic compounds that bacteria as well requires. It's a growth factor, like vitamins, amino acids, purines, and pyrimides. So some bacteria may require specific vitamins to grow, like biotin, for instance, or certain amino acids. So those were the chemical requirements in terms of elements and nutrients. Now for oxygen requirements. So if a bacteria cannot live without oxygen, that would be an obligate aerobe. Remember what I put there. It must have, obligate. Some people, again, just to differentiate between obligate and facultative. Some people cannot live without a marriage mate or a mate, and some people prefer to be married, but they don't find the right mate, so they can stay single while they find their mate. So this will be facultative versus obligate. So in the case of obligate aerobes, they require oxygen. They must have oxygen. Without oxygen, they don't live. Facultative anaerobes, they prefer oxygen, but can live without oxygen. They grow best in the presence of oxygen. Obligate anaerobes cannot live in the presence of oxygen. So we will see some examples of each of these groups. And air tolerant, they grow best in the presence of oxygen, in the absence of oxygen. Air tolerant anaerobes most likely are called air tolerants. They grow best without oxygen, but can grow in the presence of oxygen. That's why they are air tolerant. And then we have some that grows in the presence of small amounts of oxygen, like 5%. These will be the microaerophiles. So this is how they grow in test tubes with media. So for obligate aerobes, so if you have media here and you put these obligate aerobes, they will grow on the top of the media. Why is this, that the obligate aerobes grows on the top of the media? it's here. Just going to slap somewhere. You can read there an explanation of growth patterns. Okay, someone please read this. Louder.
Growth occurs only where high concentrations of oxygen have diffused into the medium.
Growth occurs where high concentration of oxygen is present, right? So where in the media is the high concentration of oxygen? Here, right? Because there is oxygen here. So that's where these grows, these obligate arrows. Now about for facultative anaerobes. So again, these will be bacteria that prefer to grow in the presence of oxygen, but can grow in the absence of oxygen. So it can grow best here where you have more oxygen, but as you move away from the surface, you have less oxygen, but still can grow. Now, there is an actual explanation for why this happens. Like for instance, in the case of these obligate aerobes, they have enzymes like catalase, superoxide dismutase that can neutralize toxic forms of oxygen. And for this, it has also the same enzymes, but it can let these toxic forms of oxygen to be neutralized partially and still be able to grow. Now, for the obligate anaerobes, why do they grow in the bottom of the tube? I'm going to compare you to aerobes and anaerobes. So let's say that a professor intimidates you and the professor is near to the whiteboard. Where would you sit? So you will be an obligate anaerobe. Where would you sit? In the back of the room. If you were an obligate anaerobe, if the professor doesn't intimidate you, you will sit close to the professor. And I know that happens. And not only to me, I saw it yesterday to someone else, and I was relieved because I said, well, thankfully, I'm not the only person who seems to be like a mean person, but I'm not. Okay? It's just that you're shy. But I don't bite, I don't do anything. So anyway, so this is where you have less oxygen. That's why it grows here. Now, this one, why it grows there? Because it lacks enzymes to neutralize harmful forms of oxygen. So while these ones have enzymes that can detoxify oxygen forms that are toxic, these ones don't. That's why it grows here. And then for air-tolerant anaerobes, so this one can grow evenly throughout the media because oxygen has no effect. So it has only one enzyme, like superoxide dismutase, that allows this oxygen to be partially neutralized and helps them to tolerate oxygen. And now for these microaerophiles, grows where they have 5% of oxygen, which will be a little bit away. So in here, for instance, we have like around 21%, which is like the amount of oxygen that we have in the environment. And then further away, it's like 5%. So this one produces lethal amounts of toxic forms of oxygen, and if it's exposed to normal atmospheric oxygen, will produce more toxic forms of oxygen, and then it will die. So this is the reason why this one's growing here. This one's growing everywhere. This one's only on the bottom. This one on the top, and this one's better in the bottom, less in the rest of the tube, but still can grow anywhere. So let me give you an example before we end. Like, we have a bacteria, it's called Mycobacterium tuberculosis. This Mycobacterium tuberculosis can infect our lung, can produce lung tuberculosis. And then our lungs, they have this conical shape. So this is the apex, and this is the base of the lungs. So Mycobacterium tuberculosis it's an obligate arrow. And there is more oxygen here in the apex, so the concentration of oxygen is greater here than in here. And this is O2, sorry. So the infections produced by Mycobacterium tuberculosis, you expect them to be more at the apex or more at the base, based on the fact that it's an obligate arrow. So more at the apex will be number 1, more at the base will be number 2. Let's see who does what I do. Will it grows more at the top of the test tube or at the bottom of the test tube, because it's an obligate arrow? And some of you already put the finger, that's correct. The rest who haven't done it, show me. Is number 1 grows better at the apex or grow better at the base, number 2? Here, this is where it creates a lot of cavities. It destroys, it creates gaps in our lungs in the apex, because that's where it has more oxygen. That's where it's not get killed. So if you ever see x-rays of a person who's coughing a lot, has very thin weight, and then it has in its lungs cavities in the apex, more than likely these x-rays come from a person who has tuberculosis, lung tuberculosis. Okay, so that's all for today.